Hi, ladies and gentlemen. It is Ms. Corcoran, and we are going to talk about respiration today, chapter 7, and we're going to be drawing. Actually, I have 4 pieces of paper, so we're going to be drawing 4 different pictures about respiration. And I know this is really the overview and the introduction, and I strongly recommend you come back to this at the end as a review. So the first time we go through it, you're going to be like, oh my gosh, this is awful, I don't like it. But I think it will be a wonderful review tool as well. So the first thing we're going to do is we're going to draw the chloroplast, the mitochondria. Okay, and just make sure you know all the parts. I'm going to draw this one kind of big. Okay, here's your mitochondria. That's your mitochondria, and there's your outer membrane. It's a double membrane organelle. The inner membrane is bent and folded, and the more bends and folds it has, the more surface area it has for ETCs. If you recall, during photosynthesis in the thylakoid membrane, that was the location of the ETC. And I wanted you to remember that ETC equals ATP. And the more membrane you have, the more ETCs you can have, which means the more ATP you can make. So this is your inner membrane. Okay. And these folds, of course, are called the cristae. So the folds are the cristae. This right here is the matrix. And this is your inner membrane space. Okay, it's important that you can visualize this because this space right here is where we're going to be pushing all those protons or hydrogen ions, actively transporting them into the inner membrane space, and then they're going to fall back through, back into the matrix. Okay, so that's our first drawing, one and done. Let's move on to the next one. So cellular respiration occurs really in 3 parts, and I'm just going to do a really quick overview. The first part, I'm going to draw smaller mitochondria. Okay, so the first part, part 1, is called glycolysis. And glycolysis occurs in the cytoplasm. Once again, this is your mitochondria. Okay, parts 2 and 3 actually occur in the mitochondria if oxygen is present. We have something called the citric acid cycle. That's going to occur in the matrix. So that would be step 2. And then step 3, I'm going to put a big fat X and label that the ETC, electron transport chain. But one of the things we're going to do in cellular respiration is refer to it as oxidative phosphorylation. That's an oxidative, that's a giant V, excuse me. Phosphorylation. Okay, so this is your overview. You've got glycolysis, citric acid cycle in the matrix, X, and then your oxidative phosphorylation in the ETC. Okay, now let's talk about each part. Step 1. Glycolysis literally means splitting sugar. So I want us to keep up with the number of carbons, kind of how we did in the Calvin cycle, just because you had to regenerate that RUBP and you needed to know how you got back to 5 carbon RUBP when 1 carbon Carbon dioxide is fixed during the Calvin cycle. And please don't look at my fingernails. My gosh, I'm going to get my nails done as soon as I'm done recording these. So glycolysis means splitting sugar. So we have 6 carbon glucose. It is going to be split into 2 molecules of pyruvic acid. They each have 3 carbons. Pyruvic acid can also be called pyruvate. So don't let that confuse you. During this process, it's really like a 10-step process. And lucky for us, we are not responsible for knowing all of those 10 steps and all of the enzymes that catalyze those 10 reactions. It's kind of messy, and we don't have to know that. What do we need to know? We need to know that we are oxidizing glucose. What does it mean to be oxidized? Well, it means that we're going to rip some electrons off. And we have an electron carrier in this process called NAD+. And NAD+ is going to take an electron from glucose, and it's going to turn into NADH. Okay, so we have reduced NAD+ when we oxidize glucose. And NADH is an electron carrier and has one job. NADH's job is to deliver the electron all the way over here to the ETC. Okay, if and only if oxygen is present. In the process, we are also going to create 2 ATP by substrate level phosphorylation. And I'm just writing net because technically speaking, we create 4 ATP, but we have to invest 2. So all I want you to know is that when glucose is oxidized during glycolysis, we make pyruvic acid, which has 3 carbons apiece. We make 2 ATP, which isn't very much. That's why I want you to know the number, by substrate level phosphorylation. And we reduce NAD plus to NADH. And if oxygen is present, NADH is going to go to the ETC. Boom. That's all you need to know about glycolysis. That's pretty cool because it's a really complicated set of reactions, and that's all you need to know. No oxygen required. And it occurs in the cytoplasm. Okay, so it occurs in the cytoplasm, no oxygen's required. We're going from a 6-carbon molecule to 2 3-carbon molecules of pyruvic acid. In the meantime, we create an electron carrier that's going to carry electrons to the ETC, and we create a teeny tiny bit of ATP. I take it back, friends. You needed 5 pieces of paper because we have 2 more things. So now we have our pyruvic acid, our pyruvate. So we have 3 carbons of pyruvate, and it's going to go into the mitochondria. So I'm going to draw our mitochondria, but I'm only going to draw it sort of like halfway because I need to draw it kind of big. That's our mitochondria. All right, so our pyruvic acid goes in there. There's another step right here before we start the citric acid cycle where pyruvate gets oxidized. Remember, we have 3 carbons. We're going to lose a carbon in the process or in the form of carbon dioxide. So we've gone from 3 carbon to 2 carbon. And we're also going to rip off some electrons and create NADH. Okay, we're going to add a coenzyme. And we're going to end up with something called acetyl-CoA, which has 2 carbons. This right here we call the oxidation of pyruvate. It would be step 1.5. Remember glycolysis is number 1. This would be step 1.5. And then step 2 is the citric acid cycle. So we have our 2-carbon acetyl-CoA, and it is going to combine with something called oxaloacetate. Oxaloacetate. I cannot spell that without sounding out all of the syllables. That has 4 carbons. That's part of the cycle. Acetyl-CoA is going to join that. What is 2 plus 4? It is 6. We're going to make citric acid, which has 6 carbons. All right. Again, another set of complicated reactions and intermediates and a whole bunch of enzymes that catalyze all of the reactions from one to the other. But all you need to know is that this joins with this to form this. And then we need to get back to this. Okay, we call that a cycle. So if you have 6 carbons and you want to go to 4 carbons, what are you going to do with some carbons? That's right. You're going to get rid of them by breathing out carbon dioxide. Perfect. We are also going to oxidize the heck out of this molecule. And when we oxidize it, we're going to reduce something else. So we're going to reduce our NAD plus to NADH. We're going to create another one called FADH2. All right. NADH, FADH2, and the NADH, excuse me, that should not have a plus there. I beg your pardon. That should not have a plus. Where do they all go? They're all electron carriers. They're all going to go to the electron transport chain. So this is the citric acid cycle. And this is step 2. Okay, so step 1 is glycolysis, which we already drew over here. And this is what you need to know about it. Okay, you need to know that's produced, and that's produced. Our pyruvate gets oxidized in step 1.5. It turns into acetyl-CoA, which is 2 carbons, which enters the citric acid cycle. It's going to combine with 4-carbon oxaloacetate to create 6-carbon citric acid. There are a whole bunch of molecules between citric acid and regenerating oxaloacetate. You don't need to know them. What do you need to know? That in order to get back to 4-carbon oxaloacetate, we have to get rid of some carbon dioxide, and we're going to oxidize these molecules by creating our electron carriers and reducing NAD plus and FAD to NADH and FADH2. That is step 2. So step 3 is our ETC, oxidative phosphorylation. Okay, so we have our NADH and our FADH2. What are they carrying? They are carrying electrons. All right, so I'm going to draw the, let's see. This is my membrane. And remember, it's bent. And there's my beautiful ATP synthase. So here's an up close of our inner membrane. This right here is the matrix. This is the inner membrane space. Let me show you from our big drawing. Remember the matrix and our inner membrane space. So what I've basically done is I am doing an enlargement of that right there. Ready? Ta-da, there it is, super up close. All right, so our NADH and our FADH2 deliver the electrons to the ETC. So they give the electrons. And then those electrons go wee down the ETC, okay? While they're doing that, they are going to be pumping protons into the inner membrane space. So that it is chock full of protons. Remember, this is our inner membrane space, and we are filling it up with protons. Those electrons are being pulled by oxygen right here. Remember, oxygen is super electronegative. So those electrons are so excited. They've been delivered by our electron carriers. They're going to go wee down the ETC to the final electron acceptor, oxygen, who's also going to grab some hydrogens. Because remember, if we're pumping hydrogen ions, then there are hydrogens around. So two of them are grabbed by the oxygen. And then guess what? This is going to leave as water. Metabolic water that we just created. We literally made water. So the very last thing we did is use oxygen to make water when we oxidize the sugars made in photosynthesis. The very first thing we did when we made those sugars in photosynthesis is we split water to release oxygen at PS1 in the thylakoid membrane. And I think that's really cool. All right, so we've got all these protons in the inner membrane space. Remember, they're shoved all up in here and they're building up in the inner membrane space, and they don't like being near each other. So they're going to go from high to low, and they're going to flow out through ATP synthase, and they're going to turn the wheel, and they are going to take ADP, add a phosphate, and create ATP. A whole ton of it, okay? So that is step 3, oxidative phosphorylation, chemiosmosis. I'll go ahead and put that down there as well. Chemiosmosis refers to the term of these protons flowing from high to low. Osmosis means water technically going from high to low, but we're going to say chemiosmosis are these protons going from high to low through ATP synthase. Oxidative phosphorylation is when the electrons get reduced, oxidized, reduced, oxidized, reduced, oxidized, reduced, oxidized, reduced, oxidized, and finally they reduce oxygen to make water. Okay, that is the overall. Once again, I would strongly recommend you review this at the end of the first PowerPoint lesson about respiration, because I think having gone through the diagrams in the book and the overall lesson, drawing these again will make it concrete in your head. As ever, if you have a question, let me know.